Hello everyone. So today I'm going to be showing you how we can use our SpotMap HCP analysis software combined with your 2D images to actually identify the host cell proteins that your antibody is detecting. Hello everyone. So today I'm going to be showing you how we can use our SpotMap host cell protein analysis software to actually identify the host cell proteins that you're detecting using your 2D electrophoresis gel and Western blot and your anti-host cell protein antibodies. Now, a lot of people don't know that you can actually perform identification using this technique, and that's often one of the benefits that's pushed by people using mass spec is that, oh, but we identify our proteins and we identify the peptides that we get out. And whilst that is true to an extent, the same is also possible using 2D techniques. And it works using the same mechanism. It works based on the molecular weight and the isoelectric point of the protein. And in fact, when people are saying, oh, well, we can identify the peptides and the proteins within our mixture using mass spec, that's not completely true. So when you're performing a host cell protein analysis using a mass spec machine, what you're doing is you're breaking your protein down into, all these tiny peptides and then trying to detect them, work based on their molecular weight and isoelectric point, and then work that back to say, we think the likelihood is that these peptides were from this protein. It's an analogy would be arriving at the scene of a car crash, trying to find all the pieces of a car, and then trying to work out what cars were involved in that crash. And it's kind of a situation where you're working with a best guess scenario. This gets even more difficult depending on the databases that you're using for identification. If you're using a novel cell line, something like an insect cell line to develop vaccines, the libraries that people use to compare their results to are often incomplete and haven't been validated, and there's a lot of evidence out there to show that depending on what kind of machine you use and what kind of library you compare your results to, you can get vastly different results for your kind of host cell protein identification. So this is not a facility that is unique to mass spec machines, and the gold standard for work in this area is to use orthogonal techniques, so 2D, mass spec, HCP ELISA, use all those kind of things in tandem to really get the full story of what's going on within your bioprocess. So I'm going to show you how we can go ahead and identify some of the host cell proteins that we've picked up using our anti-host cell protein antibody here in this experiment. So what we can do is, if we've included a molecular weight marker and an isoelectric gradient strip within our experiment, we can work out those two metrics from our protein spot and then use that, compare that to a library to try and get an identification for our host cell protein and get some more information as to how potentially dangerous this host cell protein could be. So the first step is to come over here to our molecular weight. Now I'm aware in these images I don't have a molecular weight marker or a isoelectric strip marker along the top, but this is just to show you how I would do it if I did have that that information as part of my experiment. So the first thing I need to do is tell the software where my molecular weight markers are and what they relate to in terms of size. So if you can save presets, so you can create a molecular weight marker, and if you're using that within all of your experiments, save it as a preset to allow you to more quickly apply it to your experiments in the future. However, by default, SpotMap does not come with any presets built in. So what you want to do is, the first thing you want to do is add some values. Now, this position here is percentage along your gel or blot, whatever, your image. So by default, we've got a 0.1 and 0.9%, and the length of the molecular weight marker is defined by these numbers here. So this means that the start means that the molecular weight ladder starts 50 pixels down from the top of the image, and it ends 250 pixels down from the top of the image. Now, if I extend this, you can see I'm getting much greater coverage of my image. And I can start adding in more different. So I can start adding more steps with the molecular weight marker defined here. So I can put this as 100. I can put this as, say, 250. I can put this as you can see, I'm starting to build up. So it's shouted at me there because I've got bad ordering. 75, 50. So this, on the right hand side, the value is where you tell the software what the molecular weight is at that point. And position percentage tells you how far along the entire molecular weight ladder that particular step is. Now you can perform this manually. So if you come across, you can click and drag and move your molecular weight ladders to match up with the molecular weight ladder that you will be able to see in your image. So you can add as many as you want. So as you can see, that is how you would apply your molecular weight ladder to your image. So as you can see, I'm just clicking and dragging around here. So we have got, you can enter the numbers manually, and you can also left click and drag when you see your cursor turn into an arrow and move them to match the molecular weight markers that will be present on your gel or your blot image when you have them. So if I come off and close that now, you can see the lines have disappeared. However, your molecular weight marker is now, you can see your numbers here. So you've still got all of your information for your molecular weight marker. And the isoelectric marker also works in a similar way. We've got presets. Now, SpotMap does come with presets for your isoelectric point. So let's just pop in. So if I put in a linear 4 to 7, I can save it, I can give it a name. I can add, again, various steps in. And I can click and drag that across my image to line up with the isoelectric strip that I would have. So once we've given SpotMap all of the information it needs to work out the isoelectric point and the molecular weight of any of the spots in our image, we can then take that information and try and work out what host cell protein that is. So as you can see, now I've given SpotMap my two ladders. As I move my cursor around, here in the bottom right you can see the isoelectric point and molecular weight information of the spots that I'm looking at. So I can either click one and get the information for that spot, or just hover my cursor over one. Now, I'm aware these two images are not aligned, they're specifically not aligned, but if they were aligned, these spots would obviously be in the same place. So we can take that information directly from the display, or we can export a report that contains this information for every single spot that we have on our image. So to do this, we come to the data access menu, and and come to results and make sure that we've got all measurements turned on. So we can copy and paste all of the measurements into Excel from here, or we can generate a PDF report. So I've actually generated the PDF report from this experiment already, and this is what you get out. So you get every single spot measurement, and over to the right hand side here you get the molecular weight and the isoelectric point alongside the spot number. Now, I don't have spot numbers turned on, but you can turn on spot numbers so you can see them in the image and try and line up your spots with your molecular weight marker. So, now you've got these two pieces of information, you need to compare it to a library to try and work out what host cell protein this, you have. Once you have the isoelectric point and molecular weight of the proteins that you're interested in or concerned about, you can then take that information and compare it to a database. So this is exactly how mass spec identification works as well. Normally the databases are built into the software that comes with the mass spec machine, whereas we obviously don't have any built into our SpotMap software, so you have the freedom to kind of take it to whatever database you want to look at. I like using the BioPhorum host cell protein database because it's searchable, and it also gives you information regarding the risk level of a particular host cell protein, along with academic papers linked to how they've come to that conclusion. So as you can see, if you just access the raw database, now this resource is open to everyone, I'll include a link in the description below so that you can access it and look up your molecular weight and isoelectric points of your proteins. So you can search it using those two parameters, or the name, etc. And you can filter it by just looking for high-risk host cell proteins. But if you want to come and look at the whole database, you can see that here you've got the high-risk host cell proteins are highlighted in red. You've also got the UniProt accession number, so you can get more information on the particular host cell proteins that you're looking up. And you can really get access to all of the information you could possibly want, cell localization, any particular notes associated with this protein. And you can also come across to the read more section, and it will give you academic papers that highlight things about this host cell protein, whether it would be their function or when they were first identified, or any characterization information. But it gives you further reading resources for you to kind of make judgment on whether or not this is something that is going to be of huge concern, that you need to rework your kind of process and your workflow to make sure you get rid of these host cell proteins, or if for your uses it's going to be kind of an acceptable risk. But yeah, you can be more informed about the host cell protein that you've identified and make your decisions from there. As ever, thanks for watching. And if you'd be interested in trialing a copy of our SpotMap HCP analysis software in your lab with your own data, please check out the links in the description below.